In 1865 to 1866, Gregor Johann Mendel published his work on pea plant and heredity. But at that time, his work did not accepted until 1900s. In 1900, three botanists, Hugo de Vries, Carl Correns, and Schermack, independently performed experiment that Mendel was also performed, and they concluded that same thing or same laws according to the Mendel. At that time, the work of Mendel has been hugely accepted and well appreciated. Hello friends, welcome to Biology Concern. I am Animesh, and in this video I am going to discuss about principle of dominance. Gregor Johann Mendel observed some characteristic features regarding the heredity of pea plants and make three laws. The first law is principle of segregation, the second law is law of independent assortment, and the third one is principle of dominance. And ultimately Mendel find out the reason behind the heredity. Mendel concluded that each of the trait that is expressed on pea plants is controlled by a particular hereditary factor, or also known as the alleles or genes. So Mendel first performed on true breeding plants. So what are the true breeding plants? In case of true breeding plant, the plants are actually, when self-fertilized, they produce identical offsprings. So this is the true breeding plant. So here is the definition of true breeding plant. A true breeding plant is one of that when self-fertilized, that means when it is self-fertilized only produces offspring with the same trait. That means if I cross a tall plant with another tall plant that is also a true breeding plant, both are the true breeding, in the next generation I will observe 100% of the plants are tall. Okay, so this is the phenomenon of true breeding plants. So basically they are genetically identical, so they are genetically identical and have identical alleles for the specific traits. The alleles are these types of organisms are known as homozygous. So the alleles are homozygous in conditions in the case of true breeding plant. The breeding experiment of Mendel is that when Mendel crossed a tall plant that contains T and T, those T and T stands for the alleles. Okay, so if this is a gamete and these are the chromosomes, right? So these are the chromosomes. So the allele should be capital T and in the other chromosome it should be capital T. So both of the chromosome consist identical alleles. So these alleles are in homozygous condition. So basically these alleles are in homozygous condition. And in case of short plant, if this is a gamete and if this is a chromosome, this is the pair of chromosome, so the allele should be small t and small t. That means these chromosomes represents the short phenotype and these chromosomes represents the tall phenotype. Whenever in parental generation, tall plants are crossed with short plants in F1 progeny. So F1 means first filial generation. So in F1 generation we get 100% tall plants. So how can I conclude that the 100% plants are tall? So this phenomenon observed only in Punnett square. So what is basically Punnett square? In 1917, Reginald C. Punnett, an English geneticist, discovered a process or invented a process that helps to identify the gamete number and the percentage of the gamete. So basically a Punnett square is constructed by drawing a grid. So basically it is a drawing of grid. Each cell contains an allele. So basically each of the cell contains an allele from each of the corresponding gametes and generating the genotype of the progeny. So it ultimately generates the genotypes of the progeny. So if this is the Punnett square, so each of the box contains an allele. So this is the allele, this is the also allele, this is also allele, and this is also allele. So whenever we crossed capital T to capital T with small t, that is the chromosome, so we get capital T small t, capital T small t, capital T small t, and capital T small t, and one box represents 25%, because the four box represents the 100% phenotypes or 100% offsprings. So one box represents 25%. So in this case, in the first manner, we get 100% tall plants. Okay. So this is the first breeding experiment of Mendel. So in this case, capital T is dominant over small t. That means whenever this capital T present, that means whenever the chromosome that is represents the capital T. So if this is a heterozygous condition and one of the allele is capital T and other one is small t, so then capital T masks the action of small t. That means capital T dominate small t and ultimately inhibit the expression of small t and express its own phenotype trait, that is the capital T or tall. So capital T is dominate over small t. Whenever this capital T present in single dose means single number of allele, then it can suppress the action of small t and ultimately produces its own phenotype expression, that is the tall. Okay, so in F2 generation we get, when self-fertilized F1 into F1, we get 75% of the tall and 25% of the short. Just observe this Punnett square and you will observe this very interestingly, that this capital T here small t, capital T small t. So we got capital T to a 25% case, we get capital T small t in 25 plus 25, that is the 50%. So if we plus 25 plus 25 and we get small t and small t 25%. So in this case 25% is homozygous dominant, 50% is heterozygous, and 25% is homozygous recessive. In case of the recessive plant or recessive trait, that is the small t or small t, whenever this kind of phenomenon expressing, then they are present in double dose. Okay, that means they are in homozygous condition, because in this case if capital T small t present, then capital T suppress the action of small t and small t does not present or does not express, then we do not get any phenotypic expression of small t. But whenever both of the alleles are small t, then this small t and this small t both are expressing and we get the small phenotype of the plant. So wherever a recessive character is expressed, it should be in the double dose manner. Mendel concluded that because the short trait reappears in the second generation, here you observe there is a no short trait. So here no short trait appears only in F2 generations. So there is a no short phenotype expressed in F1, but in F2 generation we get a short phenotype expression. So the trait for the short plant was not lost during the crossing over. This is the first conclusion of Mendel. That means the small t does not lost in the crossing over of F1 generation, and each of the trait is given plant is controlled by two hereditary factor. As I told earlier that two genes are present, that means two corresponding chromosomes are there and two alleles are there. So this is the one hereditary factor and this is the second hereditary factor, one and second. Okay, so in this case one and second, the first and second hereditary factor are there. And in the F1 generation, the hereditary factor of the tall plant, that is the capital T, seem to mask the expression of hereditary factor of the short plant, that is a small t. So in the conclusion, we can now conclude that capital T whenever present, then it masks or suppress the expression of small t, and if short a phenotype is expressed, then it should be in double dose manner. Okay, so it should be in double dose condition. So whenever capital T is present, capital T dominates over small t and ultimately mask the expression of small t and ultimately express itself. So the capital T in this case is dominant and small t is recessive. Okay, so small t is recessive. So this is the principle of dominance, where one character or one allele is dominate over another allele, and these kind of things ultimately express themselves. That means this kind of alleles, dominant alleles, express themselves and they mask or suppress the expression of the recessive trait or recessive allele. And this is the principle of dominance given by Mendel. I hope this video is helpful for you to understand the principle of dominance in Mendelian genetics. If you like the video, please hit the like button. Don't forget to subscribe, and thanks for watching.